Welcome back. In this lecture, I want to describe the spreading and thriving, sadly, of tumors. Previously, I talked about how tumors can be benign or malignant, and how a physician will diagnose and stage a tumor, and tumor grading as well. Then I began talking about how tumors grow, and the fact that they grow rapidly at first and then rather slowly later on. In this lecture, I'm going to describe the most feared property of cancer, metastasis, and the spread of tumor in the body. Then I'll describe angiogenesis, how a tumor recruits its own blood supply, building on the story of Judah Folkman and his hypothesis in the previous lecture. Finally, I'll describe the tumor burden on the body. Metastasis is the most feared aspect of cancer. Tumors can grow, and maybe a surgeon will be able to take it out, but once the tumor spreads, it's a very bad prognosis. About 60% of tumors have metastasized at the time of diagnosis. And as I mentioned before when we talked about tumor staging, a tumor with metastases at the time of diagnosis is, in most instances, incurable. We can control it, but we can't cure it. Metastasis, the spread of tumor at distant sites in the body, is related to angiogenesis, the recruitment of a blood supply. You'll see the similarities as we talk. Metastasis comes from the Greek met, which means change, and histonai, which means to stand in place. So let's do a little story of metastasis. A man sees his doctor with pain in the back and an elbow. And the doctor gives the man, the usual drugs and the painkillers, etc. A week or two later, the pain is still there. So the doctor says, okay, there's bone pain, let's do an x-ray. So it might be a break or something like this. The x-ray shows nothing. Pain's still there. Now the doctor thinks, well, there might be something else going on. There might be some damage to the bone. Now when bone gets damaged in some way, bone tries to repair itself, and the way bone repairs itself is it recruits calcium, which is a major element, as you know, in bones. It recruits calcium from the blood. So what the physician can do is to replace calcium with a radioactive element called technetium. It looks like calcium, it acts like calcium, it's not calcium, but it's radioactive. So if bone is being repaired for some reason, technetium may show bone repair, and here's what happened with that man. What it showed was spots on the elbow of bone repair, extensive repair, spots on the back. And those spots were metastases due to what turned out to be prostate cancer. The prostate cancer cells had spread, that is, they had gone through the blood or lymphatics to the bone, where they landed, and they burrowed into the bone and were causing damage there, and the bone was trying to repair itself, and that's where the technetium was taken up. The mystery of metastases really was something that people had puzzled over for centuries, until the early 1800s. In 1829, French physician Jean Recamier first showed in animals and in humans that metastases occur from a primary tumor breaking off small cells, and those enter the bloodstream or other circulatory system, and then seed themselves onto other organs. Before, it was thought that metastases were just new tumors. They were arising spontaneously somehow. Metastases occurs in a series of steps, and I will come back to this repeatedly throughout this lecture. In the first step, tumor cells grow toward a blood vessel. Tumors aren't sitting by blood vessels all the time. They might be several cells away. There might be angiogenesis, which little small capillaries, but tumors have to grow toward a significantly sized blood vessel. So tumor cells will grow toward the blood vessel. In the second step, some tumor cells will enter the blood vessel. They'll sneak their way in through the wall of the blood vessel. If it's a small blood vessel, it's called a capillary. Third, the tumor cells are carried in the blood circulation. Fourth, they stop somewhere. They arrest, right? The tumor goes off and they stop somewhere in a target tissue. Fifth, they leave the blood at that point, and then they grow in the target tissue. That's a metastasis. So now I'm going to describe these steps by which the tumor metastases happens. And I'm going to start by by describing tumor cells growing toward the blood vessel. So how does that happen? Here's a tumor invading and growing toward a blood vessel. And as the tumor invades and grows toward a blood vessel, I have to emphasize it's an active process. The tumor itself makes enzymes or proteins called proteases. Now, the way enzymes are named is the term at the end is ACE, A-S-E. An ACE is something that breaks down something or catalyzes something. A protease is something that breaks down proteins. So at the leading edge of the tumor, if my head is the leading edge, are these enzymes being spewed out, and they're digesting the extracellular material outside of the cell. Because if a tumor is going to grow toward a blood vessel, it's got to eat its way toward the blood vessel. And it literally eats its way through adjacent normal cells, etc. Tumor invasion is an active process. And the actual events are that a tumor cell makes these extracellular proteins called proteolysis, proteolysis, breaking down, lysis is breaking down something. And then the tumor essentially migrates, right? The leading edge of the tumor kind of burrows its way through. There is a name for these particular enzymes that the tumor makes. They're called matrix metalloproteases. Matrix metalloproteases. We'll call them MMPs. These MMPs digest the matrix, the collagen and other things outside the cell, as well as the other cells in the nearby area. And only the cells at the leading edge of the tumor make these MMPs. And as I said, it's active. It's not just pressure of the tumor as it grows, but it's kind of creeping its way like an alien in a movie, and it's creeping its way toward the blood vessel. And that's why you see in a malignant tumor, as I showed in the last lecture, a fuzzy edge. And that fuzzy edge is the edge that's kind of growing its way toward a blood vessel. The movement of those cells, the cells just sit there unless something in the cytoskeleton tells them to move. So the movement is by internal cytoskeleton fibers, much like a muscle, and it kind of contracts and moves the cell along. Well, there are genes normally that stop cells from moving. Those genes are anti-metastasis genes. They control this. There's a protein called collapsin. It's a protein that inhibits cell movement by binding to the cytoskeleton and keeps cells in place. So it's not surprising when you see a Kaplan-Meier curve that looks like this. Here is the probability of disease-free or survival. Disease-free, in other words, whether the disease is not there, and then of course, as you go down, the disease is there. And this is in lung cancer, in gene expression. And we find that lung cancers that have a high level of expression of this inhibitor to cell movement have a better disease progression than ones that don't have it. In other words, if we can stop cells from moving, we can very well stop metastases and tumor growth because the tumor can't grow toward the blood vessel. This is an active area of research. The MMPs have all been characterized. There are people making drugs against the MMPs. There are people making better molecules than collapsin to prevent cells from moving. So understanding all of this, and I have to emphasize this throughout the course, that understanding all of these steps, all the science behind cancer, is leading, as we speak, to therapies. Well, the next step 2, is that tumor cells are going to enter the blood vessel. Now, I have to step back a minute and say, well, how is the tumor going to enter a blood vessel? The blood vessel has a lining, right? That's number 1. And number 2, the tumor is a ball of cells. A cell has to say, bye-bye, parent tumor, got to leave now, have to leave home, and go into this blood vessel. So 2 problems here. Well, first of all, getting into the blood vessel, that's done by the MMPs. They're enzymes, and they'll poke a hole in the blood vessel wall, and they'll allow the tumor cell to sneak in. Tumor cell detachment, well, we alluded to this in the last lecture. Remember, I talked about a membrane protein that adheres a cell to the matrix outside the cell, to the collagen fibers, etc. And I mentioned that in a number of, in tumors typically, there's a mutation. DNA change, caused by a carcinogen, of course, and the DNA change makes the protein that doesn't attach well. And of course, that detachment is going to allow a tumor to leave the parent, and especially if that detachment involves these cell recognition proteins that bind one cell to another. So remodeling of the cell adhesion proteins or attachment proteins allows the tumor cell to leave and go into the blood vessel. So now we have a tumor cell that has just entered the blood vessel. Let's go back to the cancer metastases overview. I've gone to the first two steps. The tumor cell has grown toward the blood vessel. The tumor has grown. The tumor cells are now in the blood vessel. We're now on step 3. The tumor cells are carried in a blood, or I'll mention in a moment, lymphatic vessel. What is a lymphatic vessel? There's a lot of fluid around tissues, especially epithelial tissues, and that fluid can build up to very harmful levels. And so we have a system of pipes or tubes in the cell called lymphatics that essentially drains the fluid. It's especially true at the ends of blood capillaries, capillary beds. And so to prevent fluid accumulation, there's a lymphatic system that kind of parallels the circulatory system, the arteries and the veins. And the lymphatic system carries that fluid back to the heart, eventually. So lymph vessels drain tissues, as well as blood vessels draining tissues. Typically, carcinomas on epithelial cells are drained by lymphatics. And so if a tumor is going to spread, it will spread in lymph vessels before it enters the bloodstream. In a sarcoma, which is inside and right next to a blood vessel, they'll spread by the blood. But that's not a hard and fast rule. What happens then is millions of tumor cells can be shed daily into the blood and lymph. Take a look at my hand here. You'll see a small tumor that's about 1 centimeter in size. That's about the limit of size that you might detect. It may not be causing a problem, may come up in a CT scan, whatever. It's a small tumor. That's got 1 billion cells. 1 billion cells. So imagine this next to a blood vessel, and it'll release a couple of thousand, a couple of 100,000, whatever, a day, okay, as it grows. Typically, this is a dangerous trip for a cell in the blood. Typically, only 1 cell in 10,000 survives. But you know, if a tumor is releasing millions of them, there's enough that are going to survive to be a problem. In addition, at this stage, when they're in the blood, the tumor system is susceptible to the immune system. It might be rejection and attack. We'll see this later on. There's a new approach to looking at metastases and detecting tumors, by the way. If tumors are in the blood, if tumor cells are in the blood, let's see if we can detect them on the way. And so technology is being developed quite rapidly now to detect small numbers of tumor cells in the blood. You can spread some blood over the suitable matrix, and the blood, the tumor cells will adhere to this matrix because they have certain exterior proteins and normal cells will not. So that's a good indication of metastases, right? That the potential to metastases is there if we can detect tumor cells in the blood. That is a developing technology. Okay, so now the tumor cells in the blood, and now we're at step 4. It stops. What do you mean it stops? Circulation is going all over the body. What makes a tumor cell stop at a certain spot? It's got to be at the exterior of the cell, a cell recognition phenomenon. It's got to be a protein sticking out of the cell that says, I recognize this organ as a place to stop. Want proof? Let's take a cell, tumor cell, and let's remove all the cell contents and just have the membrane. Right? No DNA, no nothing. We just have the membrane. If you put the membrane in to the blood circulation, that tumor cell will stop at its normal metastatic place. So the membrane is the place where that label is that essentially is arresting at a certain spot. Now, sometimes it's a spot that's nearest in the circulation, not necessarily so. So this graphic shows you where sites of metastases are. For example, in brain, the primary tumor that arrests there is lung and sometimes breast and melanoma, renal cell, which is kidney, colorectal, right? On the other hand, pancreatic cancer very often goes to the liver, right? Prostate cancer very often goes to the bone. This is a cell recognition phenomenon, not just because it happens to be nearby. Next step, the tumor is arrested. Now it's going to leave the blood vessel and enter the target tissue. How's it going to do that? The same way it got in. It's going to make proteases, these MMPs, poke a hole in the blood capillary. It's already bound there because it knows where it is, and it's going to burrow its way out into the tissue now, into the new organ. Step 6, the tumor grows in target tissue. Well, this is obviously the reverse of what we saw before, right? So the tumor, it's going to grow in a distant organ. Instead of a big tumor making a small piece, now it's a small cell that's going to grow and go into a big group of cells. The result is pretty sobering. This is a photograph post-mortem of a liver cell with metastases from pancreatic cancer. The liver, of course, is the dark brown, and there are the metastases. Metastases can be a disaster because they're, in this case, of course, they're going to horribly compromise the function of the liver. Angiogenesis. How does that work? Again, it's recruiting at a metastases or a primary tumor. Angiogenesis is recruiting a blood supply. Now recall, a tumor cell or a cell in the body has to be no more than 5 cells away from the blood in order to get a decent exchange of gases and nutrients. Now, I want to emphasize, what do I mean by that? Exchange of gases and nutrients. Oxygen is being released from hemoglobin and it goes to a cell. Now, oxygen being released from hemoglobin will enter a cell that's right there, right by the blood capillary. And it does this by a process called diffusion. Diffusion is a process, for example, if you take a drop of ink and you put it in a glass of water, the ink molecules are all concentrated in one place, then you diffuse, come back a couple of minutes later, and the ink is evenly diffused. If someone enters the studio here who has a lot of perfume on, DNA perfume, the DNA perfume is concentrated in the person, it wafts through the air toward me. That's diffusion. Diffusion is a slow process. So the farther away a cell is from the source of oxygen, namely the blood, the less likely it is to get oxygen in a timely way. And so that's why cells have to be pretty close to the source of oxygen, 5 cells away. And that's the importance of tumor angiogenesis. So what are the steps in this? You're going to be reminded of some of these steps from the study of metastases. There's a small localized tumor here. There's a nearby blood vessel here. The tumor sends a signal to the blood vessel. We've talked about this briefly before. The signal goes to the blood vessel, and the blood vessel makes a branch. What do we mean? A couple of things have to happen. The signal will land on the cells of the blood vessel, and those cells are called endothelium. It's just a technical term for the lining of the capillary. And it stimulates the cell wall, the muscles of the lining of the cells that line the blood vessel. It stimulates those cells to divide. And those cells will divide, so you'll get more cells that line a capillary. And now the cells have to migrate. How are they going to migrate? They're going to migrate up by digesting their way using MMPs, proteases. They're also going to migrate up because they can move, because they've got a cytoskeleton. Very similar to metastasis, isn't it? And then they'll form a blood vessel. And the blood vessel then will form the circulatory system for a growing metastasis or tumor itself when a tumor begins. Of course, this has to be characterized, and I mentioned earlier on when I talked about Judah Folkman's work that it has been characterized. Now I'm going to name what it is, and we'll talk about a little bit about it clinically. The signal, there are a number of signals that actually come from a tumor and diffuse down to the nearby blood vessel. But the major one is called VEGF. VEGF, vascular, blood, endothelial, remember that, the endothelium is the lining of the blood capillary, growth factor, VEGF. Now, normally, the VEGF system is working all the time to restore oxygen supply to tissues when the blood circulation isn't adequate to nourish tissues. For example, in an embryo, when new organs are forming, the new organ doesn't have enough oxygen because it's so far away from the blood, and the lack of oxygen stimulates the synthesis of VEGF, and the VEGF then goes to the nearby blood vessel and stimulates it. When you exercise a lot, if I'm in the gym and I exercise a lot and my muscles start developing, those muscles, as they grow, are going to need a blood supply. Initially, the muscles are starving of oxygen. They need more oxygen, and so they make VEGF and they signal more blood vessels. And you've probably seen pictures of bodybuilders with all the blood vessel is very, very well developed. So low oxygen stimulates VEGF. Same thing in a tumor. Remember tumor dormancy from Folkman. Low oxygen there stimulated the signal, and the signal is VEGF. VEGF then goes down to a receptor protein, the VEGF receptor, which is on the endothelium lining the blood vessel. And a series of signaling events happens, and those signaling events lead to the molecular events of the tumor, of the blood capillary, making a branch and cell division happening. As you can imagine, the ability to do VEGF and do all this signaling is very important in the progress of a cancer. So here is a graphic of a Kaplan-Meier curve for VEGF expression in lung cancer. If there is low VEGF expression, the tumor can't recruit a blood supply really well. Well, that's good news in terms of tumor growth, isn't it? Because a tumor may grow just to a limited size and won't be able to recruit a blood supply and grow any bigger. So that's good, not much angiogenesis, and so survival is better. Unfortunately, most tumors are really good at making VEGF. And in fact, sometimes there is a mutation in the promoter, which is the region that involves, that promotes gene expression. There's a mutation in a region that promotes gene expression that will make it more active and make more VEGF and make this a much more sensitive system. And so you'll get better, you'll get better expression of VEGF, and so you'll get a recruitment of the blood supply. So metastasis and angiogenesis are really to be feared. In both instances, I cannot emphasize too much that the science we know is leading to therapies that can be, that are being devised. Tumors are a burden on the body. That should be obvious to you. As tumors grow, they affect functions of the body, but at the same time, the body responds to the tumor. The body does everything it can to reject a tumor. And the rejection of a tumor is done by a system called the immune system. Now let me briefly introduce the immune system. The immune system is a system in the body that recognizes things that are foreign to the body. Foreign things might be an invading pathogen, it might be a virus, it could be a cell that's different, like a cancer cell. So recognition is a major function of the immune system. It's done by certain white blood cells. The second thing that happens is mobilization. Once the recognition event happens, the white blood cell says, oh, a foreigner. And essentially sends a signal out to other white blood cells that says, get the army going. And so the army gets going, and they make things you may be familiar with called antibodies, which can fight disease pathogens, that can lead to the organism being killed, or they can make cells that will kill, called killer cells, that can kill the target cell. Now it turns out that tumor cells make molecules that are unusual. Think of the very first story I told you of Kareem Abdul-Jabbar, the basketball player. He had a leukemia that made a fusion protein, a brand new protein, that's only made in that leukemic cell. It's made nowhere else. So if the body has a way of recognizing that cell, the immune system is going to say, gotcha. And here are the events of the gotcha would be. So the tumor would be recognized by white blood cells. The white blood cells then send a signal and expand their numbers. Send a signal to various soldiers in the white blood cell system that are somewhat different, that will then home in on any tumor cell that has that marker and kill it. Great. Here's the problem. The problem is, number one, tumor cells can be so large in number that they will overwhelm the immune system. And number two, they may not have the new protein or the new molecule they are making in a way, in an exposed way, that the immune system can see it. If you look back on the graphic, you'll see that it appears to me that the white blood cells are recognizing this little triangle thing on the outside of the tumor cell. Now, in the case of the lymphoma, for example, this is turns out to be a kinase, protein kinase that's targeted by Gleevec. It's inside the cell. So we got a problem. This thing is inside the cell. Immune system can't see inside the cell. It can't see by the cell membrane. It doesn't have any periscopes or something. So it can't see inside the cell. It can only see that stuff that is presented to it. So immune presentation is a very important phenomenon in immune system killing cells by these natural killer cells. As you will see later on in the course when I discuss this as a new therapy, there's a whole lot of science going on to try to get the tumor to coax the tumor into exposing its new proteins to the immune system. And once those proteins are exposed, then essentially the immune system will take over and kill the tumor cell. A corollary of what I've said about the immune system attacking tumors, and it does in many instances wipe out tumors at a very early stage, and we never see them, is that people who are immunodeficient, like people who have AIDS, for example, a disease where it's an immunodeficient, their immune system is not working well, are much more susceptible to cancer. And there are a number of cancers that are characteristic of AIDS patients. Unfortunately, tumors may evade this killing system. Here is a cell, tumor cell being killed by white blood cells. And unfortunately, the proteins may not be exposed, so now the tumor is going to continue to grow. This is where the tumor burden becomes an issue. And a tumor burden means how big and how harmful is the tumor to the person who is carrying it. Tumor burden is a disastrous consequence. And as the tumor grows, it releases substances and may block organs as it's getting bigger, organ obstruction. It'll release proteins that are being harmful. Blood vessels will burst in the tumor as it's growing and release blood in the stool, etc. Hormones can be made in the tumor to upset the functioning of the body as well. As the tumor progresses, there is a condition that develops in many cancer patients toward the end of their lives called cachexia. It's a Greek term which means bad condition of the body, and it is indeed bad condition. In cancer cachexia, the tumor itself is releasing substances into the blood that are going to various places. For example, in the fat, it's breaking down fat. The fatty acids are going to the liver, and they're deranging the chemistry of the body. Skeletal muscle breaks down. So muscle wastage happens. The muscle proteins break down to amino acids. They also go to the liver. So it's a kind of disastrous thing. It's almost untreatable. About a third of all cancer deaths are due to cachexia. The other 2/3 are due primarily to organ obstruction or the tumor making chemicals that essentially damage organs in some other way. So physical obstruction or other ways of death. In cancer cachexia, a number of things are contributing to it. I described some of them here. Abdominal pain, clinical depression, which happens in many patients, constipation, which is common and a side effect not only of the tumor, but of the treatments for cancer, obstructing the intestine so bad if a tumor is in that area. Side effects of the drugs and the radiation that are used. We'll talk about this later on in the course, what those side effects are. Very often because of cachexia, because the tumor is releasing substances that go to the brain and the tongue, there are taste alterations, so people don't want to eat, and malabsorption of food. And overall, a derangement of energy metabolism, so the person becomes quite weak. In the end, there is what's called a wastage, and the person with cancer will lose lean body mass, and this graphic says essentially that the more lean body mass you lose, at 10%, there's an impaired immunity, person can get infected more. If you lose lean body mass, that's not if you're obese, but if you're a lean person, if you lose 20% of it, there's decreased healing and weakness, and by 40%, they'll be largely due to infectious disease, but also other causes of pneumonia and blood clotting. In this series of lectures, the last 2 lectures, I've described tumor growth. And I've described how tumors can be benign or malignant and how they're diagnosed. Something about tumor growth as it grows rapidly and then slowly. In this lecture, I described metastasis and angiogenesis, the spread of tumors, and the recruitment of the blood supply. And finally, I described tumors being a burden on the body. So we've essentially taken a cancer from an initiation of mutations in DNA to changes in the cell, to the growth and development of a tumor, to the spread of disease, and sadly, in too many instances, to mortality. Now we turn to the greatest achievement in the war on cancer, I think, a description of what exactly goes wrong inside a cancer cell at the molecular level, and this is where we have great hope for specific diagnoses and treatment. Thank you.